Hey, hello everybody, and welcome to the very first Biomech-V webinar. This is meant to be a series of online biomechanics lectures. Today, our speaker is going to be Adam Shortland. I'm Dan Bassett, and just going to take a few minutes, not even a few minutes, but just a few seconds here at the beginning to give you an idea of what Biomech-V is supposed to be. It's kind of been a dream of mine for a few years now, and I have to acknowledge, first of all, Richard Baker, who has been a big supporter, and to be honest, this wouldn't be happening right now if Richard Baker hadn't helped make it a reality. So the idea of it is that it be a free resource to spread high-quality biomechanics lectures to anybody that has access to YouTube. It's been great that the International Society of Biomechanics has chosen to endorse this, and I really want to thank our first speaker, Adam Shortland, who has generously volunteered his time to bring a talk. I've met him at a few different conferences, most recently right here in Italy at ESMAC, and I heard an excellent talk. And just to give you a little bit of a background on him, he's had 20 years of experience in muscle. He did his PhD in imaging using ultrasound, a postdoc in the mechanics of large muscles, and in '98 he started running the Gait Lab at Guy's Hospital in London. And from this experience, he's been able to do some excellent research. And I don't really want to take any more time. I'm going to pass the word directly to Adam.
Dan, thank you. Yeah, thanks to Dan, and thanks to Richard as well. It's a real honor to be here at the beginning of this enterprise. Today I'm going to describe what we require of our muscles and neurology to maintain function through the lifespan, and then we're going to look at how some of the requirements of typically developing neuromuscular system are altered in cerebral palsy. My interest arises in this area because I work with children with cerebral palsy in my day job and as a clinical gait analyst, assessing their gait and function. Writing reports, discussing treatment options with my surgical colleagues. But I can't move on without acknowledging all the parties to which I'm associated. I work at the One Small Step Gait Laboratory, which is part of the Evelina Children's Hospital, which is itself part of Guy's and St Thomas's Hospital Trust, which in turn is a partner within a strategic alliance of academic and clinical partners called King's Health Partners. I'm afraid this is the way of the world, this sort of stuff. So anyway, let's move on. I actually am situated, like I say, at Guy's Hospital, which is in central London, which is the tallest tallest hospital building in Europe. There used to be a girls' hospital as well, but that was closed due to government cutbacks. Guy's Hospital, like I say, is the tallest hospital in Europe. That's not here. That's this one, and I actually work in the basement, so I don't get much of a view. A central theme in today's talk is the determinants of function across the lifespan, explained, I hope, in terms of our nervous and muscular systems. To start, we need a model of the function through the lifespan explained by these properties. If we think about it, typically we're born at 0 years of age, and we last till about our 8th or 9th decade. And during that time, we have functional tasks to complete in our daily lives, which we need to execute properly to lead a fuller life. For example, a sit-to-stand maneuver. So what we've got here is a graph which has age along the x-axis, some sort of vague concept of neuromuscular strength and control on the y-axis, and we have a threshold, the sit-to-stand threshold, which we need to exceed to perform the sit-to-stand exercise. So at 0 years of age, We really don't do much, and we can't sit to stand. But maybe at about 12 months of age, we develop enough neuromuscular strength and control to exceed the sit to stand threshold and to be able to sit to stand. We carry on developing our strength and control until we reach a point where we develop a reserve in muscular strength. I like to call this the functional reserve. It's all good and great until about 25 years of age, when we begin to lose some of our muscle fibers, some of our motor units perhaps. So we gradually get a little bit weaker. So we gradually decline over the next few decades until we reach about the age of 60, where we start to decline more sharply. Until we reach a point, maybe in our 7th or 8th decade, where we can no longer sit to stand, say, independently of armrests. And then, after that, I'm afraid, we can no longer sit to stand. So, if you like, this is a concept of function to the lifespan based on our reasoning of neuromuscular strength and control. So, this talk, we're going to talk about what is required of our system to develop such a functional reserve, our prerequisites, if you like. We're going to look at neuromuscular correlates, neuromuscular capacity and cerebral palsy, and what causes a reduction in the functional reserve of those individuals. And we're going to examine the factors which influence our early development of neuromuscular function and how that might influence our neuromuscular function in this disease condition. And we're going to think about what implications that has for treating children with cerebral palsy. In case you don't know, children with cerebral palsy are children who have an injury to their to the motor parts of their brain, maybe prenatally, maybe perinatally, maybe in 1 or 2 years postnatally, and that affects their movement control. But as we'll come to see, it also affects their ability to develop muscle and to develop their sensory function. So, the determinants of muscular capacity. Well, you can't start off thinking about the determinants of muscular capacity without thinking first of all about the sarcomere. Is the fact that the sarcomere is the only is the basic unit of force generation within the muscle. And the amount of force that it can develop is dependent on the overlap between the thin and thick filaments in the muscle, here indicated in red and blue. The thick filament, the myosin filament, and the thin filament being the actin filament. These filaments have places on them where they can attach, where they actively attach. They form cross bridges. And it's the formation of these cross bridges that make the sarcomere generate force. Consequently, when you have maximum overlap between the actin and the myosin filament, we develop the most force. And when we have a stretched sarcomere, like in position A here, we develop very little force. This gives rise to a force length diagram, which looks a bit like that. One of the interesting things about this diagram is that we produce peak forces around 2.5 microns, and that's true across the animal kingdom, more or less. And what's also interesting is not only do we produce peak forces there, but also that our resting length, the resting length of the sarcomere, is also about at that position. That's true for all of us, within reason. I've never really thought about it much until recently, but I wondered what would be the mechanical consequences if our resting length was displaced from our peak active force generation. That you can imagine that there might be some consequence, and I'd like you to think about it, because we're going to come back to it later and discuss that, because there are some clinical implications. But sarcomeres really don't do much on their own. So we need sarcomeres to be arranged in groups to produce significant amounts of force and to produce significant amounts of velocity. So here we have a muscle. It's not a very big muscle. It's got 12 sarcomeres, 4 in parallel, 3 in series. And this arrangement of sarcomeres would produce a force length curve, maybe similar to the one you see on the right-hand side there. Let's rearrange those sarcomeres so that we have 3 in parallel and 4 in series. Now this sarcomere, this muscle, even though it's got the same number of sarcomeres, actually would have a very different force profile, because the force that this muscle can produce, or any muscle can produce, is effectively proportionate to the number of sarcomeres that act in parallel, and the velocity that it can produce is proportionate to the number of sarcomeres that effectively act in series. So these two muscles produce different force length profiles merely by their arrangement. Well, a string of sarcomeres on their own isn't that useful to us unless you have an ability to activate them, and we have nerves for that. We have nerves which interact with our muscle fibers. They interact at a location called the neuromuscular junction, and it's activation of the nerve terminus that gives rise to release of acetylcholine into that junction, which activates the sarcolemma, and by a series of chemical processes causes the contraction of this series of sarcomeres. Well, that's all very good, but one thing you should know is that we don't start out by just having one neuromuscular junction per muscle fiber. In our infancy, we may have had many neuromuscular junctions associated with a single muscle fiber, and it's the competition of the nerves for acetylcholine receptors that exist on the sarcolemma, the cell membrane, which causes a single neuromuscular junction to gather all the acetylcholine receptors together and the other neuromuscular junctions to wither away. You can imagine the deficits that we might have if we tried to activate a single muscle fiber by many neuromuscular junctions. So we've managed to activate our muscle. What else do we need of a muscle? We've got this string of sarcomeres at the moment. But as they stand, if these sarcomeres were relaxed, there is nothing connecting. There's no cross bridges here. There's nothing connecting the myosin and the actin, which means that if you try to pull this sarcomere apart, you could easily. So we need something that binds the two free bits of the sarcomere together. And what we have is this large molecule called titin. It's one of the biggest molecules in the body, and it connects the Z disc here. That's illustrated as this little black line, and the myosin molecules. And this, I suppose, helps to provide mechanical integrity. And mechanical integrity is, of course, very important to muscles. Helps to provide mechanical integrity of sarcomeres acting in series. So that's in series, but we don't just have series of sarcomeres. We have them arranged also in parallel, as you've seen. Like this is two sarcomeres arranged in parallel. So how do we maintain mechanical integrity in parallel? Well, we've got a particular arrangement of molecules called alpha-actinin molecules within our Z discs, which keep our Z disc connected one to the other. You can also see that titin and actin connects into this matrix. So now we have bound our serial mechanical integrity, if you like, with our parallel mechanical integrity. And what the parallel bits do is they hold sarcomeres essentially at the same length. So they're operating at the same points on their force length curve. That's very useful. So we've bound these sarcomeres now together. So now we've got a big block of sarcomeres bound in series and in parallel. So they're nearly a functional group, but not quite. We also, in our muscles, have connective tissue, the extracellular matrix. And here's an electron microscope. It's an image that commonly people extract from Trotter and Perslow to illustrate the point. And this is where the myofibrillar content of a muscle has been digested, and it's just left the connective tissue matrix. And what I hope you see is this this highly connective matrix. Our muscle fibers don't exist in isolation with extracellular matrix surrounding each and every one. Rather, all muscle fibers are connected by their extracellular matrix. But we need something to connect our Z disks, which we know are bound nicely, to our extracellular matrix. And that is by a series, a complex of molecules, which link into the Z disk, find their way through the sarcolemma, through the basal lamina, and connect into the connective tissue matrix. You may recognize some of the molecules involved. Desmin, I think you might recognize the name of. Then dystrophin also. And we know from our understanding of how disrupted muscles are in diseases like muscular dystrophy, the importance of having these molecules, or these molecules in non-mutated form, connecting our Z disk through to our extracellular matrix. Because otherwise, if we didn't have these molecules functioning, this matrix would be disrupted with nothing connecting our connective tissue matrix to our myofibrillar content, if you like. So mechanical integrity is maintained by this high connectivity, both serially and in parallel, between our sarcomeres and our extracellular matrix. And then, of course, our extracellular matrix itself forms our tendon, which connects to a bone, so the whole thing is linked from sarcomere to bone. Yeah, let's go through that. Sarcomeres are bound in parallel and series, as we've seen, in series by titin, in parallel by alpha-actinin. They're also bound to the extracellular matrix by this complex array of molecules, including dystrophin and desmin. The extracellular matrix is continuous around all the fibers, and that probably provides some mechanical support. It's much stiffer than passive muscle fibers, and it probably has a role in distributing the load across the muscle. And it almost certainly has a role in regulating sarcomere length. That's those alignment of those Z disks together by the alpha-actinin. And the extracellular matrix, as I've said, at least the perimycelium, is continuous with the internal and external tendons of a muscle. So we've gone from sarcomere to whole muscle, and we've mechanically connected it all. So we're making progress. We've identified the ways in which sarcomeres are bound together, but how are oh no, hold on, next slide. So we've discussed the mechanics, right? So we need now to turn our attention to how muscles are activated. To do that, we should describe the cells which activate our muscles. These are alpha motor neurons, and they reside in the grey matter of our spinal cord. They activate a number of fibers simultaneously. So they control the activation of a number of neuromuscular junctions. And the number of neuromuscular junctions, the number of fibers that they control per alpha motor neuron, is called the innervation ratio. And the collection of the alpha motor neuron and the fibers which it activates is called the alpha motor unit. Now you'll notice something rather special about the distribution of the fibers that this alpha motor neuron activates within the muscle. That they're spread out. They're not neighboring each other. That's interesting, especially when you add another alpha motor neuron with its own territory. Now here you see that the alpha motor units are overlapping. And again, this probably allows or dissipates stress concentration within the muscle. So it distributes the tensile load by letting motor unit territories cover much of the whole cross-section of a muscle. Motor neurons also behave in a stereotypical fashion when activated voluntarily, that is, from the motor cortex. When a pool of alpha motor neurons are activated, it is the smaller motor units that are activated first. Smaller motor units arise from smaller alpha motor neurons. And as we increase our descending input into our pool of alpha motor neurons, we start to activate larger and larger alpha motor neurons and larger alpha motor unit territories. This principle of activation by size is called the Henneman principle. And it's an interesting one because it means that when we activate our muscles, we activate them with the small ones first, the small units first, because many of the activities we do in our daily lives are not forceful activities. So why would we activate the big, costly motor unit territories when we can activate smaller motor units to generate small forces? Indeed, we only activate the bigger units when we need more explosive actions. Interesting too that the order of relaxation of these units is the larger motor units first. So they turn off first when we de-recruit our alpha motor neurons. We've covered that alpha motor neurons activate groups of fibers, and collectively they're called alpha motor units, and also that their territories are rather large in the muscle. They cover quite a cross-section. An individual alpha motor neuron will innervate fibers through quite a cross-section. And we've also covered the Henneman principle, where descending input from the brain activates smaller motor units at first, and then as that descending input increases, it activates larger and larger motor units, and that's very helpful in the control of our muscles. We also have descending inputs that act on the alpha motor neuron, inhibitory inputs that act on the alpha motor neurons, which depress their activity, as well as having segmentally derived signals from the muscle, from other tissues, that also feed and may activate the alpha motor neurons themselves. As well as all this, we have cells that aren't alpha motor neurons within the spinal cord grey matter. These may be referred to as interneurons. They form networks of cells which may influence the activity of the alpha motor neuron. For example, when you're activating your dorsiflexors, these cells may help you to inhibit the activation of your plantar flexors. This act is called reciprocal inhibition. These cells may also be the origin of central pattern generators, those low-level central nervous system circuits that are responsible for rhythmical actions in our body, like swallowing or indeed walking or running. These are very important. If we want to have rhythmical, nicely defined activation of agonist-antagonist as we walk, then these cellular networks need to be well developed. We're also talking about, for a muscle to be functional, for a muscle to create this reserve, it needs to adapt to the demands placed upon it, and it does that by synthesizing or degrading proteins. So it responds to mechanical and electrical stimuli, tensile forces within the muscle, or activation of a muscle, but it may also respond to nutritional factors like leucine being present in the blood supply, and other circulatory factors like the presence of insulin-like growth factor or tumor necrosis factor. And these influence protein synthesis and degradation. It's actually quite interesting to look inside the cell. And if we look inside, here's a very simplistic view of a cell. We've got sarcoplasm, which is the cellular fluid in the muscle fiber. We've also got one of the nuclei within the muscle fiber. The muscle fibers are actually multinucleic. And we've got the sarcomere. So we're interested in how sarcomeric proteins are synthesized and degraded. And the regulation of that occurs by the presence of what are called transcription factors. These are proteins that are attached to a gene within the DNA which regulate the output of that gene. So there are transcription factors which promote synthesis of proteins, sarcomeric proteins, and there are factors, these ones in purple in this screen, which may limit the synthesis of proteins or even excite the pathways which degrade sarcomeric proteins. So these transcriptional factors are very important in our muscle adaptation. They are also influenced by other factors, other proteins, enzymes, etc., that may exist within our sarcoplasm that have an influence on the transcription factors. Until recently, we didn't know that much about how mechanical stimuli. Well, let's talk first about the circulatory factors. As well as these transcription factors, we have circulating factors, like I said, insulin-like growth factor, tumor necrosis factor, TNF, and also myostatin that are present in our circulation to different levels. Now, IGF-1, for example, actually stimulates the green side, the hypertrophic side of our transcription factors. And that gives rise to the development of sarcomeric proteins. Whereas myostatin and tumor necrosis factor upregulate the degradation side, or at least limit synthesis. I didn't know until recently that, and in fact nobody knew until recently, I don't think, and it's 5 years ago, that about the mechanism for which, the precise mechanism for which we increase our sarcomeric proteins by way of adaptation to mechanical stimuli. And that's because these and so the story is that these transcription factors actually don't just reside in the nuclei, but they get transported between the nuclei and in the largest part the Z disc within the sarcomeric structure. So when sarcomeres are mechanically activated, they release the transcriptional factors that are bound to the Z disc into the sarcoplasm, and these are taken up by the nucleus and they stimulate the production of sarcomeric proteins. Equally, during the activation of a muscle fiber, calcium is released into the sarcoplasm. And this calcium, the presence of calcium, stimulates a factor called calcineurin, which activates a series of transcriptional factors within the nucleus, which again leads to the building of protein. So, in a very simplistic way, this is how we respond to mechanical stimuli and develop protein. As well as adaptability, we need to respond to injury when we get it to our muscles. This is a micrograph of a muscle fiber that you're seeing in the center of the screen there. And you can see little blue dots, and they are myonuclei. So they are close to the surface, close to the sarcolemma, and they're distributed throughout the fiber length because fibers are very thin and long, and so you need many nuclei to express protein and degrade protein regionally. The yellow cell is of interest. It actually resides outside the sarcolemma, outside the cell. It's called a satellite cell, and let's have a more simplistic diagram. So here we have a cell again, muscle fiber, and we have the blue myonuclei, and then we have this satellite cell. When we have a trauma to that cell, there's some damage, we get activation of the satellite cells, proliferation of them, and not only do we get proliferation, we get migration to the site of injury. These satellite cells form myoblasts, sort of muscle cell precursors really, and they bind to the site of the trauma, and they donate their nuclei to the muscle fiber in order to repair it, so that a muscle fiber is reformed by this increase in myonuclei. So let's go over the summary of those prerequisites. For a muscle to operate optimally, we need a muscle to have optimal sarcomeric function. we need to have a certain morphology. If we want a powerful extensor, we need lots of sarcomeres orientated in parallel. We need mechanical integrity, and we went through how sarcomeres were bound to the extracellular matrix. We need control over those muscles, and that's delivered directly to the muscle by the alpha motor neuron, but that's mediated by signals from our periphery, signals from our cortex, our motor cortex, and signals, rhythmical signals that are generated within the cellular network in the grey matter. We need adaptability, so we need to respond to demand, and when things go really wrong, we need to be able to repair those muscles. So let's now think about how these prerequisites might be altered in a condition like cerebral palsy. Cerebral palsy, like I said, is a condition in which you have a neural injury that affects your corticospinal tract, and obviously it's a neurological condition, but because it affects your corticospinal tract, it also affects the development of your muscles and then ultimately your bones. So how when we have an injury like that, how are our prerequisites altered, these muscle properties altered? Why is it that children and adults with cerebral palsy have less of a functional reserve than their typically developing peers? We're going to look at that period in kind of middle life, if you like, after year one, where they've developed their motor milestones, maybe a bit later than year one, and we're going to look at that mid portion of life when they're able to maybe perform these functions like sit to stand. And we're going to look at this group. So generally, when in the next few slides, I'm talking about individuals between the ages of about 6 or 7 and about 23. So the first thing to talk about is resting sarcomere length. As we said before, sarcomere length is pretty stable across species. So it was an extraordinary finding of the Lieber group, Smith being the first author here, to find that in a group of children with CP, ambulant children with CP who were going for surgery to lengthen their hamstrings, they took samples of the tissue from their semimembranosus, I think, and their gracilis, and they compared to reported sarcomere lengths in typically developing individuals. And what they found was that the children with cerebral palsy had resting sarcomere lengths of 3.6 micrometers, which is an extraordinary thing, considering that throughout the animal kingdom, around 2.5 micrometers seems to be the going rate. But what implications has that for sarcomeric and ultimately muscular function? I kind of think it's all. We often talk about children with CP as having essentially a neurological condition, which of course they do, but we often really ignore the secondary effects. But if you think about these sarcomere interactions, it explains many of the things that we would normally attribute to neurological phenomena. For example, dynamic equinus. Children with cerebral palsy often walk on their toes. So that's an interesting thing. So it's often that's often seen as just a neurological phenomenon, if you like, that toe walking. But is there a myological basis for it? So let's consider the resting length status of our child. Here is the resting length, 3.6 microns, for our child with cerebral palsy. If this muscle wants to operate at an optimal position, it must get those actin and myosin filaments to maximally overlap, so there is a maximum number of cross bridges. To do that, it must force the ankle into equinus. So, in other words, it would be natural, of course, or appropriate, I should say, that a muscle would want to operate actively in its optimal active position, i.e. when there are a maximum number of cross bridges formed. In the case of children with cerebral palsy, that means they would go from a resting state, where their ankle was near neutral, into a equinus state when walking, where their ankle was in a great deal of plantar flexion. if you think about it as well, if these muscles are sarcomeres are very stretched, it would also mean that the muscles were very slow. Because if you went from a resting position to an active position, an optimally active position, you would need to form the maximum number of cross bridges. But at the moment, you're very far away from doing that. So you would need to activate. Each cycle of activation would cause a contraction of the sarcomere by one displacement of one myosin head. So you'd have to count the number of myosin heads to get a optimally forceful contraction, because you're starting from a position of very few cross-bridge connections, and you need to be at a position of a lot of cross-bridge connections. So what else is wrong? What also is wrong? Well, we talked about muscle demand, that sarcomeres arranged in parallel are forceful muscles. So it was interesting to us when we measured muscle volume in children with cerebral palsy. These are muscle volumes from a series of muscles in the lower limb of individuals. I think these are between 10 years of age and 23 years of age. And what we've done here is we've normalized the muscle volume to the body mass of the subject to take some of the non-clinical confounding variables out of it. And what we found was that when you did that, the muscle volume per unit body mass of children with cerebral palsy in the lower limb are almost universally small compared to their typically developing peers. Some of these muscles are very small. So, for example, the soleus there in this cohort has lost about a quarter, or is about a quarter to a third less volume than their typically developing peers. That's likely to have some consequences for muscle mechanics. What's also interesting to look at is the relationship in normally developing individuals and in individuals with CP with body mass. If you reproject that data in terms of with body mass along the x-axis and muscle volume, not muscle volume by body weight, pardon me, that's just muscle volume along the y-axis. And what we've got here is a linear relationship. The open circles are the normally developing individuals and the closed squares are the children with CP. And you've seen the normally developing relationship. These are 4 of those muscles, but it could have been any of those 11 muscles on the previous slide. Is that not only is there a difference between the size of these muscles in the 2 groups, but also their trajectory of growth with body mass is different. So that has some real implications. So that means that as children with cerebral palsy grow, their muscles don't grow at the same rate that they're growing, which, if you think about it, when they reach a certain body weight, there may be a problem in maintaining their upright position. We're also interested in muscle composition. So we used MRI, a technique called Dixon imaging, to look at the fat content within the plantar flexor group, within the groups in the calf, in fact, and the tibialis anterior. And what we found was that children with bilateral cerebral palsy had greatly increased levels of intramuscular fat in those muscles. The sorts of fat levels that you'd see in obese children. One extraordinary example was the level of fat within the soleus muscle, where you had up to 16% or more of fat within the muscle. Obviously, this fat is non-myofibrillar tissue, so it's also going to compromise mechanical performance. So we've talked about muscle structure and content, and we found that there are real deficits there, both in sarcomere function and in the number of sarcomeres in parallel and the composition of the muscles. We're also interested in muscle activation. We talked about muscle activation before, and this is an experiment from Stackhouse from about 10 years ago, where they asked children to activate voluntarily their quadriceps muscles and then their triceps surae. And they had 2 groups: children, typically developing children, children with cerebral palsy. The blocks in black here are the typically developing children, and the the ones in white are the CP children, the children with CP. And what we found, or what they found, was that children with cerebral palsy have an inability to activate the muscular resources that they do have. So what this figure is saying is that if they maximally stimulated these muscles, that's a figure of 1 here, but what you can see from this is in the quadriceps femoris that children with cerebral palsy could only act about 50% of their muscular resources. And that's also similarly true of the triceps suri. So now we have a compromised muscle, poor sarcomeric function, and now an inability to selectively activate those muscles. We talked about mechanical integrity a lot, and there are also data from the group of Lieber. Here we have controls on the right hand side, and what's staining for here is connective tissue in the figure, collagen 1 and laminin on the figure on the left hand side. And what you see here is the increase in the connective tissue matrix in children with cerebral palsy, and that may lead to problems with the connectivity, as we've discussed, between the sarcomere and the connective tissue. Right, that's fine. The other thing we talked about was satellite cells, the ability of muscles to repair themselves. Also from the Lieber group, they've shown that the number of satellite cells existing within children with cerebral palsy are much lower than those from typically developing sample. Now that means that children with cerebral palsy may have an ability to repair their muscle. So what sort of muscular contractions would cause damage to a muscle? Well, we know that when we go to the gym and the gym instructor tells us to do eccentric exercises, what we're doing is we're causing a little bit of damage so that we can repair them and make the muscle bigger and better. So perhaps these children, if they perform eccentric muscle contractions, may never recover from those eccentric contractions because they have a limited capacity to do so. Some time ago we did an experiment where we attached an ultrasound probe to the calf muscle. We looked at typically developing adults, typically developing children, and 8 children who toe walked who had cerebral palsy. With the ultrasound image we could see the muscular tendinous junction of the gastrocnemius here, the muscle belly of the gastrocnemius, and this is the skin surface up here. So down here is the ankle, up here is the knee. Because we've got markers on the probe and markers on the person, we can see exactly where the muscular tendinous junction is in space at any instant in time. And what we found was, and that meant we could measure the length of the muscle belly and measure the length of the tendon. And this is data from the change in length of the muscle belly through that cycle. The little, the solid line is from typically developing adults, and the line with triangles is from typically developing children. And what I hope you see here is that during the single support phase of gait, these muscles are performing isometric contractions. You would expect them to perform eccentric contractions because, of course, your ankles are dorsiflexing in this phase of gait, so that you would think that the muscular tendinous unit is lengthening. However, this implies that the tendon is lengthening, but the muscle belly is staying the same change in length. However, that's a nice way of performing muscular contractions because that maintains isometric contractions, is probably very efficient. Also, it avoids damaging contractions. But if you look at the children with cerebral palsy who toe walked, that toe walking, perhaps, in combination with the weaker, smaller muscles as we've seen, seems to give rise to an eccentric contraction of the muscle belly during single support. This is likely to be damaging for these children. So one of the things we should be looking at is how do we avoid damaging contractions? How do we avoid making a bad muscle worse? So we talked about the deficits that these children have, but perhaps we should finally draw our attention to what the origin of these deficits are. So we're now going to look at very early infancy, and in fact prenatally also. It's interesting when you see an ultrasound scan of a fetus which is maybe only 9 weeks old, and everybody's surprised that this little fetus that they can see under the ultrasound is moving. They think, oh, we know Johnny's moving. It is an extraordinary thing, because at that stage in the development, 9 weeks post-conception, in fact, there is no connection between the motor cortex and the spinal cord. So Johnny's moving because there are spontaneous discharges from the spinal cord that are activating the muscle that is present there. So at that stage, we have no connection between cortical and spinal circuits. They are developing independently. It's not till about 17 post-conception weeks that a diffuse projection of axons from the cortex begin to form the corticospinal tract, which enters the top of the spinal cord. At this stage, there's no connection, like we said, from the cortex, but there is lots of afferent input from the muscles themselves into the spinal circuitry. There's very weak efferent output from the alpha motor neuron pool, because these alpha motor neurons and their interneurons haven't developed properly yet. We have underdeveloped spinal circuitry at this point in time, and we have underdeveloped spinal circuitry because one of the roles of our corticospinal tract is to develop these circuits, and I'll talk about that in a minute. And we have because we have very poor efferent output, we also have very poor stimulus of the muscles. So our muscles at this stage are weak and probably characterized by small diameter fibers. By the time we reach 2 years of age, a lot of things have happened. The corticospinal tract, which was at 17 weeks, was at the top of our spinal column, has now reached the bottom of our spinal column. It's activated all the alpha motor neurons between the top and the bottom by about 35 post-conception weeks, so about term. That has done several things. It's competed with the afferent input from the muscles for space on the spinal neurons. So it's eliminated branches of the afferent input from the muscles, reducing their influence on muscle activation. It's increased the efferent output by the alpha motor neuron pool, because it's enabled sustained contractions to be developed and for the alpha motor neurons to lower their threshold of activation. And it's also refined spinal circuitry within the spinal cord to improve reciprocal inhibition. So, and because it's done all these things, it's increased efferent output, which has developed our muscle fibers and made our muscles bigger. That's of course only half the story, because that's the motor side of the story, but we've also got the sensory side of the story. Probably our motor and sensory systems develop at a similar pace, and they're very dependent on each other. So if we think about a proper functioning motor system, we also need input from the periphery that our brains can process so that we can send the right next command to our muscles in the periphery. So they're very interdependent, the development of these things. We're not going to talk too much about the sensory elements. So what about cerebral palsy? We know there's an injury here. It might be a focal injury, as I've described here, illustrated here in this half brain that we've got here. We've got the cortex with all the axons forming the corticospinal tract arising from the motor cortex, and then we've got the thalamus here, and the corticospinal tract coursing through the internal capsule here. There's an injury that takes out many axons that would be present in an intact corticospinal tract. It also probably from its sequelae, prevents the myelination of those tracts. So if we consider that an injury in cerebral palsy happens around between 24 and 32 weeks gestation, typically, that's right in the middle of the development of our corticospinal tract and the jobs it does as our corticospinal tract courses through the spinal column. It's likely that that has an impact on the development of the corticospinal tract, such that the corticospinal tract will compete less well with the afferent segmental nerves for termination onto the alpha motor neurons. That in effect means that in children with cerebral palsy, we would expect greater, which is what we see, greater afferent information coming into the spinal cord, influencing muscle activation. Some people call that spasticity, increasing the reflexive component. Let's think about it in another way. That injury occurs again, it has the impact on the development of CST, and that causes a lack of refinement of those cellular networks within the cord. That means that we've no longer got as an effective way of promoting agonist behavior activation and depressing antagonist activation. We've probably got poor reciprocal inhibition if we don't have properly developed cortical networks. And that's again what we see in children with cerebral palsy, a persistence of premature patterns of activation, lots of co-activation in those children. And if the CST, the corticospinal tract, doesn't develop very well, it also may fail to lower the thresholds of activation of alpha motor neurons, which would lead to a reduction in alpha motor neuron output and failure to activate muscular resources, which, as we've seen, is also a feature of cerebral palsy. So, in conclusion, which leads to poor muscle development. So, in conclusion, we can say that we can trace back the phenomena we see in the child in the mid-years, from 6 to 23, if you like, to events that happened, particularly the development of the corticospinal tract and the injury that happened to it in very early infancy. So, what do we do about it? We have a neuromuscular problem with neuromuscular capacity. And that problem with neuromuscular capacity, if we don't develop a neuromuscular reserve, we probably have a problem with maintaining it in the long term, because even typically developing human beings decline in their neuromuscular capacity as they age, as we've discussed. But if you have a small neuromuscular functional reserve, then it's likely that you will lose your mobility earlier, because you don't have the reserve on which to lie on, to which to degrade. You're going to fall below threshold earlier than your typically developing peers. So what might we do about that? Well, one simple solution is to build neuromuscular resources through exercise. And indeed, we, among others, have shown that you can increase muscle volume in children with cerebral palsy by giving them focal muscular exercises, resistance training to build their muscle. We'd hope, we don't know, we'd hope that in the long term, that by building their muscle, developing a bit more muscular reserve, that they might be able to keep their mobility for longer. But those sorts of studies are very difficult to do. Another implication is we know that these problems arise, well, of course, prenatally, but also they persist perinatally and postnatally. Perhaps this early postnatal period of muscular and nervous development gives us a window of opportunity. This is when our brain and our spinal cord are at their most plastic, when we've not yet myelinated, created that insular layer around our nerves, which gives us more ability to adapt, to structurally adapt. But at the moment, we don't know if a properly improved and intense sensory environment might stimulate appropriate motor responses, appropriate development of the corticospinal tract. I talked before about how tied sensory and motor pathways are, and it may be that if we stimulate the sensory pathways in the right way, we could help to develop the motor side. But really, our window of opportunity is very, very early. After a few years of life, it gets increasingly difficult to alter our neurology. Finally, there are implications for assessment here. If we consider this functional reserve diagram, how do we assess it? We don't really assess functional reserve presently in clinics. We do series of functional tasks. There's a series of tasks called the gross motor function measure, which gets the child to do different activities in standing, walking, sitting, and lying, and we score them. But we score them for one task at a time. So, can you sit to stand? Okay, so in this example, this person will be able to sit to stand. But that's not really a measurement of their muscular reserve. What we should consider is doing repetitive actions, like repetitive sit to stands, so that we challenge the muscular reserve. And by doing such, we might well be able to assess the size of that muscular reserve. So, in conclusion, we have a reduced functional reserve in cerebral palsy, and I believe that that's explained by problems that these children have in their muscular capacity and in their ability to activate those muscular resources. We have seen that this neuromuscular problem is likely to be traced back to delayed development of the corticospinal tract in the early years. We thought that maybe if we might be able to improve mobility by maintaining that functional reserve in the older child, and perhaps by strengthening or other exercises, and by enhancing the sensory environment in the very young ones, which may lead to us assessing children in a better way, assessing measures of motor function that include this concept of functional reserve. And you can read more about it in an article that my colleague Martin Goff and I produced in 2012 there. If we want to talk more about imaging and how we could develop the technical side of these things, we can start thinking about how we might be able to measure muscle organization. There are techniques in MRI, for example, to measure, called tryptography, which measures essentially how water is aligned within a structure, and we can measure fat and connective tissue, as you've seen, and we should consider better ways of measuring muscle activation as well, and routinely we should measure these impairments before and after treatment. I'd like to give thanks to my colleagues, Martin Goff, orthopedic surgeon here, Nicola Fry, clinical scientist, Andrew Lewis, former PhD student, and Jonathan Noble, now Whitney. Thank you very much.
Well, thank you very much, Adam. This has been a great introduction to what Biomech-V is meant to be about. Very fascinating talk. And those of you that are watching live right now, you're welcome to leave a few questions, and we'll see what all we can get to. In the meanwhile, I'll go ahead and kick off a question. We only have time for a few, but I was particularly interested in your discussion about dynamic equinus, where children with CP we often associate as having spasticity, and we always think of that as a neurological disorder. What you presented today obviously shows that there's more going on. In fact, it seems to be more of a mechanical problem. And in no way am I discounting whether or not cerebral palsy is neurological or mechanical. Obviously, there's a major neurological element. But with regard to an equine type gait, would you say that this is primarily a mechanical problem then?
Thanks, Dan. I think what I'm trying to say there is that we as clinicians, we carry a lot of baggage with us from our historical understanding of a condition. So in this case, we've got cerebral palsy, which, is obviously considered a neurological condition, has largely been assessed and treated by neurologists. And so we've had a very neurological perspective, a bias, if you like, on the condition. What I'm really trying to say here is that because we've had that bias, we see everything about the child with cerebral palsy in neurological terms. And it's only really latterly, in recent years, that we've really started to consider it, these secondary phenomena, these secondary problems of musculoskeletal deformity, as being really very important and central to the problems that these children with cerebral palsy have. So what I'm trying to do by saying this is that these results from the Liebe group need to be verified, they need to be assessed, but certainly it makes mechanical sense that you would want to be in your optimal mechanical position to produce force when you were doing a dynamic activity. So it's possible that this dysregulation of resting and active force length gives rise to this problem. However, we don't know, and there are experiments that you could do, quite simple experiments, I think, that would absolutely demonstrate that that was the case, that there was a disconnect between resting length of the muscle fiber and the active length, if you like. I would expect that in normally developing individuals, when you watch them walking, you saw the ultrasound technique there, you could measure their muscle fibers when they were walking against the resting state in both groups, in typically developing children and in children with cerebral palsy. Then you could prove this theory, essentially, because in cerebral palsy, you could show that they shortened their fibers, and by other experiments, by stimulating their muscles appropriately, you could show that that was at the peak active force development position. And when they walked with that fiber length. Yeah. So there are experiments you can do. I think interesting little experiments with ultrasound in combination with the child walking with the ultrasound probe attached to them, and them on the couch with the ultrasound probe attached to them, and with their muscle stimulated, that you could look at the force length profiles of the active force length profiles of those children and what they do when they're walking. Does that make sense? So I don't know, but I think it's a possibility.
Yeah, that makes sense. If you want people to see, you might want to switch back to your camera.
Oh, right, okay, all right, okay, I've got to do that, yeah. Oh, how do I do that now? Hold on a second. Yeah. Just got to work out. Oh, no. It's me on. Can I just do it by...
clicking there, yes.
That's it. Oh, hi. Hello. Sorry. It's being... when you are technically idiotic, it's very difficult to control these things.
No worries.
Okay, so do we have any more questions?
At this time, we don't, but I think we're about out of time anyway. Thanks again so much. It's been really great, and look forward to more Biomech-V lectures coming up. We have Philip Rowe next month, and if you want to keep updated with any of the Biomech-V news, sign up for the newsletter. The link is in the description down below, and hope to see you all at the next lecture, and give us some feedback. Let us know what we can do better. Obviously, technology is always fun to deal with, but we'll keep trying improving. Thanks for watching, and we'll see you at the next time.